Hello class, welcome back. So for this whiteboard, we are going to do metabolism locations in the cell, everything from glucose metabolism down through pyruvate and into the Krebs cycle, fatty acid metabolism coming in from another direction, phosphocreatine and that source of energy, lactate metabolism in the Cori cycle, and even sometimes, yes, protein. Your body does cannibalize itself. That is a real thing. You can learn all about that. It's pretty disgusting, but when we run out of fuel sources, your body needs to find it somewhere. Side note, there's a whole race day fueling whiteboard session that I did that should be in this whiteboard playlist somewhere, so you can avoid doing that to your body when you exercise.
So first, of course, we have the outside world, which is basically the blood, and then here we have the cell membrane. On the inside of this, of course, is the cytoplasm, and this is where the first pathway begins. So we'll start with glucose, which we can write just like that, and that gets sent into the cell from the blood and goes down several steps, many of which we don't have the space to get into, but a big step is glucose 6-phosphate, which goes through several steps down to pyruvate, which we'll put PYR. Now, for comprehensiveness, we have hexokinase in terms of enzymes, and phosphofructokinase. Exercise training does increase these levels, and by the way, phosphofructokinase is the rate-limiting step of glycolysis. Another important enzyme here is phosphoglucomutase, which is hard to draw in, but this helps to separate pyruvate formed by glycolysis versus the storage of glycogen, where glucose 6-phosphate gets converted into glucose 1-phosphate, which then turns into UDP glucose, which then can connect to the chains of glucose-forming glycogen. Now, getting back to glycolysis, glucose 6-phosphate, instead of becoming glucose 1-phosphate, it turns into fructose 6-phosphate and goes down the steps to pyruvate. We get to a critical branch point here. In the presence of oxygen, pyruvate continues into the mitochondria via the mitochondria outer membrane and the inner membrane. Coming right in, pyruvate continues straight on through, and via pyruvate dehydrogenase gets converted into acetyl-CoA. Now, in the absence of oxygen, or if there's not enough O2, or in essence when energy demands exceed oxygen supply or its rate of use, pyruvate gets converted to lactate. The enzyme here is lactate dehydrogenase, and lactate can get sent back to the outside world and participate in the Cori cycle, going to the liver, where lactate can get converted back to pyruvate and ultimately back to glucose to be sent back to the exercising muscle for more energy. Lactate can also go to the brain, kidney, and heart. So lactate is truly not a waste product at all. Okay, let's get back to acetyl-CoA here. So pyruvate, if we're following carbon flow, glucose is a 6-carbon molecule, which gets broken up into 2 pyruvate molecules, which are 3 carbon molecules. But then when pyruvate gets converted to acetyl-CoA, acetyl-CoA is a 2-carbon molecule, which means 1 CO2 gets produced. That acetyl-CoA, in the complete oxidation of glucose, can get sent in to the Krebs cycle or the citric acid cycle, there we go, which also releases CO2. Both of the CO2 molecules get released, both of the carbons from acetyl-CoA get released. Also, we have NADH and FADH2. Sorry, that's kind of sloppy. And the final step, those molecules will send their hydrogen ions into the electron transport chain, oxidative phosphorylation, which generates plenty of ATP. Now, the one path we haven't talked about yet is fatty acid oxidation. Running out of space here, but if we have fatty acids coming from the outside world in, combines with a CoA molecule to become a fatty acyl CoA, which then can get shuttled into the mitochondria via carnitine. And carnitine is interesting because it exchanges the CoA for itself, carnitine. So inside you actually have a fatty acyl carnitine, which then re-exchanges it back here to carnitine and, once again, our acyl CoA. Now that fatty acid is also in the mitochondrial membrane, although it's getting kind of crowded here, that can undergo beta oxidation through several steps here, where 2 carbons of the long-chain fatty acid molecule get removed to form none other than acetyl-CoA. In terms of enzymes, the third step, 3-hydroxyacyl-CoA dehydrogenase, or 3-HAD, or sometimes referred to as beta-hydroxyacyl-CoA dehydrogenase. That is the enzyme that removes those 2 carbon units to form the acetyl-CoA, and that is also trainable with exercise. Well, that, and along with carnitine, even though it's not actually a enzyme, will make it red. So we have several areas in red where exercise can exert a nice training effect. And if you're interested for comprehensiveness, cytochrome oxidase in the electron transport chain, which is just going to be too small to draw in here, but there is an enzyme in there that does get upregulated with exercise training. Several enzymes get upregulated in the Krebs cycle, including oxoglutarate dehydrogenase, succinate dehydrogenase, malate dehydrogenase, citrate synthase. Citrate synthase is perhaps one of the more well-known ones, as it can go up really high, up to 150% rise in levels in trained compared to untrained people. Some of those elite marathoners have exhibited very high levels of that enzyme. And another enzyme that we didn't really get into, but in lactate there's this monocarboxylate transporter, or MCT, that helps take the lactate from the working muscle and transport it via MCTs to the brain, heart, liver, and kidneys as part of that Cori cycle, and that also gets upregulated. So we're about done here, but there is one other macronutrient that we have not mentioned that can technically participate here, which is protein. Now, it's not a preferred source, but in certain extreme conditions, such as several hours of continuous exercise, the body may run out of other sources. Unfortunately, there's no good, quote, storage form of protein in the body, as there is with glucose in the form of glycogen, or with fat in the form of, well, fat. But proteins can be used for energy if needed. Unfortunately, our body has to remove the amine group to form urea, which also leads to fluid loss, as urea must dissolve in water and leave the body as urine. It can get transaminated, where the nitrogen is donated to some other acid and forms these alpha keto acids, such as oxaloacetate, alpha ketoglutarate, which can enter into the Krebs cycle at various pathways. It can also actually turn into pyruvate as well. Some other amino acids can get converted into glutamate, succinyl-CoA, fumarate, and these can all enter into different steps in the Krebs cycle. Unfortunately, the main source of these proteins is your actual muscle tissue itself. This is why this is not a very good source of fuel, so to speak, for exercise. And in one of those extreme conditions, such as several hours of continuous exercise, muscle tissue itself cannot release glucose into the bloodstream. Right, muscle can reverse course from glycogen in this direction and go all the way up to glucose 6-phosphate, but it cannot make that last step here. So if you have some non-exercising muscle, it cannot donate, so to speak, glucose from its own glycogen stores. Therefore, the liver does that, but eventually the liver runs out of glycogen stores as well. So the only option left is that the body takes amino acids from its own muscle tissue, such as alanine. It forms that alanine from other amino acids, such as leucine, which is what is a major factor and component in muscle tissue. So up here we'll put ALA for alanine. When that gets released into the bloodstream, that can go to the liver and then finally get converted into pyruvate and then back to glucose, which then can get released in the bloodstream. That is literally called the alanine glucose cycle, and then that glucose can finally go back in. Amazingly, after about 4 hours of continuous exercise, about 45% of the liver's glucose release is not from its own glycogen anymore, but from that alanine glucose cycle. And about 10 to 15% of total energy in your body, unfortunately, is coming from muscle. So your body is literally using itself for about 10% of the energy. So our body tends to lose a lot of muscle mass if we do that kind of exercise without properly fueling up. The only other source of energy is not really a macronutrient, such as protein, fat, or glucose, but it's actually a storage form also in the cytosol or cytoplasm, called phosphocreatine. Creatine and phosphate form phosphocreatine, right? And then phosphocreatine can combine with ADP, which gets formed when our body is using a lot of ATP. Really running out of space here, sorry. I'll rewrite it down here. Creatine plus ATP can form a storage compound called phosphocreatine plus ADP. And that is a energy reservoir of high energy phosphate bonds for ATP. Creatine kinase, which again we don't have room to write that in, but that's the enzyme. It is much faster than glycolysis, but phosphocreatine has very, very limited storage here. But in situations of short-term explosive bursts, such as the 100-meter dash or weightlifting, it does not require O2 at all. As ADP builds up, as we're exerting ourselves, it drives the reaction this way and releases more ATP. So there you have it, messy but gets the job done, explaining all the different areas of metabolism and where it occurs, cytosol versus the mitochondria.